Good evening to you. Welcome back. Okay, one second. Okay, can you see the presentation on your screen? Can you see the slides, everyone? Okay, great. Hello, everyone. Welcome back. Good evening to you all. This is the week 11 of our class NPTEL Fundamentals of Protein Chemistry question answer sessions. Okay. So in today's session, we will be talking about protein-protein interactions. In the last class, we talked about protein and sugar and protein and nucleic acid interactions. But today we will be talking about protein-protein interactions. So as you understand that proteins are basically workhorses in our body. So they perform all important functions in our body. They are basically called nanoscale machines because they are tiny and they are performing all our functions. So proteins and interactions with other proteins are very important for our life processes and our everyday survival. Okay, so we'll be talking about that today. You can ask me questions if you have any questions, and if you're not able to understand something, you can ask me. If there are any doubts with the questions that I'm discussing, you can either reach out to me or you can contact the NPTEL team, and if any answer that is given is incorrect, they will help you out with that. Okay, let's begin. So first we'll talk about protein interactions. Now protein interactions refer to the molecular interactions between two or more protein molecules to carry out biological processes. For example, cell signaling, immune response, enzyme activation, structural assembly, and there are numerous examples, right? So a very common example that comes to our mind is antigen antibody interaction. Right. So this is a very common one, and similarly there are so many different kinds of protein-protein interactions. Today we will be talking about some of them. So protein-protein interactions can alter the kinetic properties of an enzyme, which may lead to subtle changes in the substrate binding and allosteric effects. Protein-protein interaction is an important mechanism for substrate channeling by moving a substrate between domains or subunits. Okay. You know about this ping pong bye-bye mechanisms, you know about multi-substrate mechanisms, and the way in which a substrate is moved between domains of a enzyme or between domains of different enzymes that are usually performed through protein-protein interactions. Then there is protein-protein interaction can help to create a novel binding site for effector molecules. Protein-protein interactions can also help in inactivating or suppressing a particular protein's activity, right? And protein-protein interactions are driven by, firstly, the shape complementarity between two domains which are interacting, and the second is definitely the interacting residues. Okay, so in an upstream or downstream events, protein-protein interactions act as regulatory contributors. So you realize how important they are, and they are basically participating in almost all kinds of cellular activity. Now these protein-protein interactions basically can be divided into different types. They can be divided based on affinity. They can be divided based on the interacting domains. Okay, homogeneity of the interacting domains. Also they can be like divided based on the timeline of the interaction. So firstly, based on affinity, there is either obligate and there is non-obligate interaction. So obligate interaction means that this particular interaction between two proteins is ultimate necessity of the two proteins to function. Otherwise, they will become inactive. Non-obligate means they are together and they may not be together based on the requirement in the cell. Then based on the stability or lifetime of the interaction. So these interactions can be either permanent, two different subunits that are always bound to one another, or these interactions can be transient. They come and bind when it is required in the cell. There can be weak and strong interactions. So we will look at these in details now. Okay. Alright, so now based on the composition of the two interacting domains, these interactions can either be homogeneous or heterogeneous. Homogeneous, you know, two domains, they are having the same sequence, same structure, they are coming and interacting. So that is homogeneous interaction. For example, you see this GROW-EL, this is a chaperone. We have learnt about chaperones in our previous classes. So GROW-EL is a chaperone which is made of 14 identical subunits, each of 57 kilodaltons, forming heptameric rings. Okay, back to back. That is homomer interaction. Then there can be heteromeric interaction between GROW-EL and GROW-ES, which make up the chaperone for protein folding. Right? And then there is obligate and non-obligate interactions. They are differentiated by affinity. So affinity means the strength of interaction with which two protein domains are interacting with one another. So an obligate interaction means that the constituents, which can be promoters. Good evening. Yes, good evening to you too. Okay. So the components of non-obligate interactions can exist independently. For instance, there is a protein called the Q protein. In this, what happens? This is involved in DNA repair. So this binds to DNA as obligate homomers. So this is an obligate interaction because this is very important for the protein-protein interaction is necessary for the function of the Q protein. Okay, coming to lifetime, there are transient and permanent interactions. The components of transient interactions associate and dissociate temporarily in vivo, while permanent interactions are very stable and irreversible. For example, interleukin 5 cytokine dimer is a permanent protein-protein interaction. So you can look at the structure of IL-5 cytokine. Alright, and see. Okay. Now, these protein-protein interactions, they can either be interactions between two different domains. There can be interactions between a domain and a particular motif. Yes, you want to say something? Yes, Ajay, you want to say something? Hemoglobin is obligate or permanent? Hemoglobin, yeah, right. The four subunits. Yes, you can search about that because we know that hemoglobin functions only when it is joined together. The alpha and beta chains, they are joined together and the tetramer is formed, only then it is functional. Right? Okay. So then you can search whether it is obligate and permanent or not. What happened to you? Okay, we'll carry on. The domain peptide complexes are mostly transient. So basically the domain and peptide. Peptide is a series of amino acids forming a motif. These often interact with one another. So I can give you one yeah. You're not understanding the difference between permanent and obligate. Okay, of course. 1 second. See, based on affinity, it is either obligate or non-obligate. And based on lifetime, it is either permanent or transient. Alright, so obligate means that two proteins cannot function if they are not bound to one another. So that is affinity. Based on affinity, they will always be bound to one another. And see here the example that they have given, an obligate interaction means that the constituents which are protomers and monomers of a complex, they are unstable on their own in vivo. So they have to be binding to one another. And what about permanent? So the components of a transient interaction associate and dissociate temporarily, while permanent interactions are very, very stable and they are irreversible. So that means the interaction is irreversible, and of course hemoglobin is having two protomers alpha beta. Yes. So in this case you can say that it is of course both obligate and permanent. But you can search, but I would say that this is definitely but is given as permanent. Okay. Yes, because hemoglobin, we don't know in nature when hemoglobin will open up, right? Hemoglobin, as soon as it is produced, it is coming together and forming this functional hemoglobin complex, and only then it is functioning as a protein. But then individual subunits of hemoglobin will not be functioning, right? That is why it is said that it is permanent interaction.
Sorry, madam, but this confusion
Hmm.
arises because madam has not said obligate or non-obligate. She has directly divided into permanent and transient.
See, these are different
That's why
ways
I
of classification. Okay, these are different ways of classification. So in
actually
this,
what is meaning by obligate is must and non-obligate means it may or may not be.
exactly, yes.
So once we say it is non-obligate, we cannot make permanent there. That's why I got confused. Sorry, madam, continue.
Okay, you can read more about this. You can find out whether it is permanent or obligate. All right.
Thank
Okay,
you.
I'll give you an example of this domain peptide interaction. Hmm. So we work with transcription factors in our lab, and we have seen this that there are certain DNA binding domains in the transcription factors. So this DNA binding domain usually binds to DNA and it is activating the gene expression. But then in the protein, you will see that besides the domain, there is an N-terminal and there are C-terminal regions. So this N-terminal region is made up of a particular sequence of amino acid which is called a specific motif. This often comes back and there is a folding and it will interact with the DNA binding domain and it will prevent interaction of this particular domain with DNA. It will reduce its affinity with DNA. Okay, so inside a protein, so many different interactions happen at the same time. Okay, based on the requirement of the cell once again. Okay, right. So let's continue. Regarding your doubt about whether hemoglobin is permanent or obligate, you can always search, and I don't think there are any questions based on this in today's class. So okay, let's carry on with the questions that we have. Okay, so this is the first question. They are asking you which of the following are correct about protein-protein interactions. So the first option is that protein-protein interface is, okay, 1 second. I missed out one slide. We have one more slide and then after that you will be answering this question, because then it will be easier for you to answer this question. Okay. So we'll go to this particular slide. Okay. We'll talk about protein-protein interfaces, interface of interaction. So protein interfaces, these are largely hydrophobic. They are also largely made up of aromatic residues. Plus, if you see, this is actually a research paper in which they are saying that histidine, you know that histidine can interact through pi-pi interaction, cation-pi interaction, right? And histidine is in kind of a dilemma at the different pH, and based on the different pH, different kinds of interactions are more pronounced. So histidine is majorly found at the interface of protein-protein interactions, and histidines interact with the aromatic residues at the protein-protein interface. So this is one very important thing that you can keep in mind. Besides that, the archetypical protein interface with defined hydrophobic core is present in only a minority of surveyed homodimeric proteins. Most homodimeric proteins are stabilized by a combination of small hydrophobic forces. And polar screen is not visible. Okay, okay, 1 second. Give me a second, please. Okay, can you see the PPT now? Okay, great. Let's continue then. Hmm. So we were talking about protein-protein interfaces. All right. So basically, the presence or absence of a hydrophobic core within these interfaces does not correlate with the specific protein functions. But then you have understood that polar interactions and interactions with water molecules, hydrophobic patches, histidine, aromatic residue interactions, these are all majorly involved in the interactions at the protein-protein interface. Okay. So now you try to answer this question. Now this should be easier for you. Please try to answer. So the question says, first option is protein-protein interface is 50% polar. Okay. So you're saying that it is C. Polar residues in the interface are usually charged. Protein-protein interactions are richer in aromatic residues. Protein-protein interfaces exhibit both shape and electrostatic complementarity. Yes, you are absolutely correct. The third option is right because polar residues in the interface are usually uncharged. Okay, the rest of these are correct. Okay, we'll go on to the next part. Now, this is the thermodynamics of protein-protein interactions. So, thermodynamics of protein-protein interactions tells you that both long-range forces and short-range forces. So, long-range interactions are represented by these dots and short-range interactions are represented by these sparse dashes. They are all important for a protein complex formation. That is all that is given here. You can read this entire thing and you know that this is the transient state, these are the intermediate states, and you can see that when there are mutants in which the long and short range interactions are removed, then the energy of both the product and the intermediate states and the transient states are rising. So their stability is decreasing. Right, so that tells you the importance of these long and short range interactions in the thermodynamics of protein-protein interactions. Okay, now we come to different techniques. So these are various different techniques, both quantitative and qualitative techniques, which are important for understanding protein-protein interactions. So please go through all of these techniques. We might have already covered most of them in our previous slides, and so most of them will be familiar to you. Some of them might not, but you can have a look at them if you're interested. All right. So we start with the thermodynamic methods. Isothermal calorimetry. This you know. And then we have microscale thermophoresis. You can read about this if you're interested. Differential scanning calorimetry, you also know. Kinetic surface plasmon resonance and isothermal calorimetry both come under these, right? Then qualitative methods. Qualitative methods, we have pull down assay, which we talked about in the last class. Immunoprecipitation, you know that here antibodies are used and you get to know the presence of your protein of interest using this. And then we have gel electrophoresis, gel filtration, and chemical cross-linking, and co-IP, that is co-immunoprecipitation, and FRET. So all of these techniques must be familiar to you. Okay. Right. So now we go to another new technique for studying protein-protein interactions. This is called far western blotting. You know that western blotting is important to study the presence of a specific protein in your sample. It helps to detect the specific protein. When we come to far western blotting, it helps to detect interaction of that specific protein with another protein. Okay, so what happens in far western blotting? Let's see. So the far western blot technique is similar to western blotting. In a western blot, an antibody is used to detect a corresponding antigen on a membrane, while in a classical far western blotting analysis, a labeled or antibody detectable bait protein is used as a probe to detect a target protein, which is the prey. So instead of antibody, there is going to be a protein which is going to bind to your protein of interest. This is the bait protein. Bait is labeled with a particular fluorescence tag, and the prey protein is the one that is there in the sample. These two are going to interact, and only when these two interact, you are going to be able to detect the signal. Okay, so the sample, usually a lysate containing the unknown prey protein, is separated by SDS, polyacrylamide gel electrophoresis, or native page, and then transferred to a membrane. The membrane is often your PVDF membrane. And then after transfer, the membrane is blocked, and then it is probed with a known bait protein. So bait comes and binds. So instead of the primary antibody, this is your bait that is coming and binding, which is usually applied in pure form. Alternatively, this reaction can be performed in a gel as described later. So the link to the website is given, and you can go through this link, and we see that following the reaction of the bait with the prey protein, a detectable system dependent upon the bait protein that is used identifies the band that corresponds to the prey protein. So the entire diagram is given here. You can go through this. So this is your different proteins that are separated based on SDS, and then there is the targeted bait protein. Bait protein is coming and binding to the prey protein. This is the prey protein. So it is binding to the prey protein on the membrane, PVDF membrane. Then what happens? Then you are adding another protein, which is actually your secondary antibody, and this is chemiluminescent substrate and HRP labeled. So it is HRP label, which is the enzyme, and then when you're adding the substrate, it is giving your chemiluminescence, and you're seeing this particular band, which is corresponding to the protein. Okay. Now we will talk about something from which you are bound to get questions in every single exam. This is the process by which antibodies are formed. Monoclonal antibodies are formed. Okay, you have to read about the process of formation of monoclonal antibodies and polyclonal antibodies. Monoclonal antibodies are usually derived by the hybridoma technology. Polyclonal antibodies are usually derived in rabbit. Okay, what is a monoclonal antibody? Let us see. So monoclonal antibodies are monospecific. That means they are binding to a definite antigen only, and they are made from the single clone of cells. And polyclonal antibodies are made up of different immune cells, and they contain antibody of different specificity. So they can bind to different antigens. Okay. So monoclonal antibodies have monovalent affinity. That means they are only specific to a particular antigenic structure, in which they bind to the same epitope. What is your epitope? This is the antibody, and these are the epitopes. So wider application in biochemistry, molecular biology, and medicine, diseases, and treatment. So they are produced by hybridoma technology. If you know hybridoma technology, brilliant. If you have forgotten hybridoma technology, or it is yet to be studied, please study, because you are going to get questions from hybridoma technology in every exam. Okay, monoclonal antibodies are widely used as diagnostic and research reagents. They are used in diagnostic kits such as ELISA, immunofluorescence, etc. So basically, what are the different applications of monoclonal antibody? They are diagnostic and research reagents, passive immunization, blood grouping, diagnosis, in cancer and imaging. So radioactive imaging. And something else is here. The entire process of hybridoma technology is given. A mouse is taken and the immunization of the mouse is happening. Then what happens? The spleen of the mouse is extracted. This is your spleen cell, and then there is screening of the this spleen cell. It is producing B cells, right? B cells specific to the particular antigen that you had introduced into the mouse. And this spleen cell, it is actually modified. And then, okay, so the spleen cell is taken and the melanoma cell is taken. So melanoma is a cancer cell and you fuse these two. And then the fused one is going to survive in the media called hat media. Okay. So, see, unfused spleen cell and fused spleen cell, these two when you combine together, they are going to give you the fused hybridoma cells, and there are different stages in which screening is happening and then selection using the hat media. So, please read what is hat media, read what is AGPRT. There are all modifications and they are all used for screening the hybridoma, which is having the property of the melanoma or the cancer cell to divide indefinitely, plus it is also producing the antigen. So after that, checking for hybridoma cells and subculturing, and then we will clone the hybridoma cells. We will propagate that in vitro or we will propagate in the mouse again, and then we will harvest it whenever it is required. This is how monoclonal antibody is produced. So please read what is AGPRT, read what is hat media, you will get asked. Okay. And why hat media, why spleen cells cannot survive in hat media, why melanoma cells alone cannot survive in hat media. That is very important. Okay. Now we will try to answer this question. Okay, please try to answer this question. Monoclonal antibodies act as modulators for targeting a large protein-protein interface. They are used in: one is screening of recombinants, treatment of many cancers, and third is diagnostic kits. For what purpose can you use monoclonal antibodies? A. Okay. A is all the three. Any other answers? Yes, you're correct. A is the right answer. So monoclonal antibodies can be used for all of these different purposes. Great. Now we come to some examples of protein-protein interactions. Okay. So one such example is the SRC protein. So this SRC is made up of different domains. The domains are SH2, SH3 and SH4. And the SH1 domain that is there, this is involved in kinase activity. Okay, this undergoes phosphorylation and it participates mainly in protein-protein interaction. Okay, so SRC is a non-receptor tyrosine kinase and it phosphorylates a variety of proteins. So this is basically a tyrosine kinase. So tyrosine kinase means that, what do you mean by a tyrosine kinase? You tell me. I'm getting any answer? Yes. Enzyme, right? Kinase means it is it is a receptor complex. Okay. But then it is saying it's a non-receptor tyrosine kinase. So then what is it? Basically what it does is, it is an enzyme, it is having this tyrosine kinase activity. So that means it is undergoing, it is basically involved in phosphorylation and involved in different cell downstream signaling events. Okay. So SRC is a non-receptor tyrosine kinase and it phosphorylates a variety of protein substrates that perform specific cellular functions. Activity of SRC is regulated by a variety of stimuli and the SRC protein is subjected to various post-translational modifications including lipidation. So if you can see the end terminal end, there is myristylation site. What is myristylation? Myristoyl group means that is a fatty acid chain, possibly 14 carbon chain. So this myristylation site is where the fatty acid chain is getting added. Besides that, there can be phosphorylation. So you see that all these tyrosines are there where phosphorylation is happening, right? Ubiquitinylation, that can also happen. Sumoylation. Sumo is a small group, small protein which is getting added to another protein. So that is called sumoylation and oxidation. So all of these different things happen, and in particular this particular tyrosine residue has been known to be an active form, while the phosphorylated this particular residue in SRC is inactive formed through autoinhibition by binding to SRC SH2 own domain. So this protein is involved in an autoinhibition. That story that I told you about this uh, transcription factors, DNA binding domain and the SLIM, that is short linear motif, where the short linear motif is coming and interacting with the DNA binding domain and preventing the DNA binding activity of the transcription factor. This entire phenomena is known as autoinhibition. So the protein is inhibiting its own activity and that is mediated by its own domain in this case. So here also we see that a particular tyrosine residue, when it is getting phosphorylated, it is interacting, it is mediating an interaction with SH2 domain, and then what happens, this SH2 domain is preventing the activity of the entire SRC protein. Okay, so this is also autoinhibition. So this is how you see that you don't need another protein to come and interact with a particular protein to perform its activity. Rather, the same protein, different domains are interacting with one another. And it is so sensitive that one tiny phosphorylation in one specific residue can mediate its function or it can interrupt its function. So SRC also plays specific roles in specific compartments of the cell, and it is localized in the membrane, cytosol, mitochondria, nucleus for specific functions, etc. Okay, right. Now we will move on to the question. Try to answer this. Cascade, can we call it? Yes, definitely we can call it. It is involved in cellular cascade. You see that here you cannot see that, but this is basically involved in cell signaling, right? But in this case you cannot see that because whatever is shown here, that is just the autoinhibition. So in this picture it is only autoinhibition. There is active and inactive SRC, and this is basically involved in cell signaling. All right. Okay. Try to answer this question. Which of the following is or are correct about SRC homology domain? One is, it is one of the two small protein binding domains found in the SRC oncoprotein. SRC is a non-receptor tyrosine kinase protein. It includes an SH2 domain, SH3 domain and tryptophan kinase domain, which is correct. Activated SRC regulates the activity of matrix metalloproteinase 9. Which one do you think is correct? Read properly. Am I getting any other answers? Okay, I've got one answer. Let's see. The right answer is C because it says tryptophan kinase domain here, and we know that this is tyrosine kinase domain, right? So, yeah. SRC homology domain is one of the two small protein binding domains found in SRC oncoprotein, and it is a non-receptor tyrosine kinase protein which is encoded in humans by SRC gene. It includes an SH2 um, domain and SH3 domain and tyrosine kinase domain. Activated SRC regulates the activity of matrix metalloproteinase 9. Alright. So option C is the right answer. Okay. Now we come to another interaction. Here we will be talking about SH2. So I think before that we can take a small break and then we can come back. Okay. So this is a 15 to 20 minutes break. Let us meet after the break. Okay. All right. See you after the break. Hello everyone, welcome back. So we are back after the break and we'll quickly finish off whatever is remaining. Okay. Okay. Yes, yes, welcome back to you too. Okay, we'll quickly finish off everything. Right. So where were we? We'll be talking about this, right? Okay. So yeah, we were talking about this. Hmm. Hello everyone. Okay, let's continue. All right. So for the next MCQ, you need to know about this another interesting thing called the phosphotyrosine binding site. Okay, so this is inactive SRC and this is active SRC. Right. Now we will be talking about the molecular mechanism of SH2 domain function. Okay, so in the inactive state, the SRC homology 2, excuse me, yeah, that is SH2 domain of SRC binds to the phosphorylated C-terminal tail, and the SH3 domain engages the SH2 kinase connector. So both SH3 and SH2 domains are rigidly coupled through a linker. Okay, so dephosphorylation of this particular tyrosine 527 or ligand engagement by the SH2 and SH3 domains melts this rigid coupling and releases the active tyrosine kinase domain to allow substrate phosphorylation. This is how the activity is initiated. Okay, so now inactive state of SHP2, this is another protein. So the inactive state of SHP2 is maintained by an interaction between the amino terminal. Okay, so whatever, from whichever source I have taken this image and the uh, writing part that is provided in your references. So please go through that, and this is possibly a review paper, so it will be easier for you to understand from there. Okay, so see, we will just go through this quickly so that we can answer the question. So simultaneously, the phosphotyrosine binding site of the amino terminal of SH2 is deformed, which impairs its ability to bind to tyrosine phosphorylated ligands. Engagement of the carboxy terminal SH2 domain by one member of a phosphorylate, uh, tandem phosphotyrosine motif is sufficient to allow binding of other phosphotyrosine motifs to the amino terminal SH2, which disrupts the association of the amino terminal SH2 with the phosphate domain and releases the catalytically competent phosphatase to dephosphorylate its substrate. So if you think this is confusing, we have the next slide to help you out. Right, so in this slide we are talking about this phosphotyrosine binding site. So we are talking about this, and you will see that this SH2 domain binds to phosphotyrosine residue within the correct context of adjacent carboxy terminal amino acids. SH2 contains a, this is important, pay attention, it contains a 7-stranded beta meander and binds with their peptide ligands that lie perpendicular to the central beta sheet. So SH2 domain couples receptor tyrosine kinase to downstream signaling pathways and participate in the regulation of cytosolic tyrosine kinase and phosphatases. Okay, now this PTB, that is phosphotyrosine binding domain, like SH2 domain, also binds to phosphotyrosine containing sequence motifs. So I told you that a domain can bind to a motif. So this phosphotyrosine binding domain, given its name, binds to phosphotyrosine containing sequence motifs within the correct context of the adjacent amino acid residue. So that means its binding depends upon the surrounding amino acid residues and their also the correct phosphorylation state or any other modification state. So some PTB domains bind to their ligands in a phospho-independent manner. PTB domains contain a 7-stranded beta sandwich with the bound peptide ligand, which is the motif, often forming an extra anti-parallel beta strand. This is important. PTB domain containing proteins commonly function as scaffolds and adapter proteins downstream from the membrane bound receptors. We will see a diagram very soon. So molecular defects in the SH2 and PTB domains that impair ligand binding are associated with human diseases, right? So yeah, this is the domain structure for SH2 and this is the domain structure for PTB. We know that SH2 is made of 7-stranded beta meander and PTB is made of 7-stranded beta sandwich. Okay, and for understanding whatever happened here in a simpler way, you need to know about these epigenetic modulators called writers and erasers and readers. So writer is a particular enzyme which comes and adds a particular, let's say, phosphorylation mark or any other post-translational modification mark. So who is coming and adding the chemical group is known as the writer. Someone who is recognizing the chemical group is called the reader, and someone who is removing the chemical group, that is called the eraser. Okay, so in this case SH2 is the reader and the tyrosine kinase mark is being added by the writer, which is the kinase, and it is being removed by the phosphotyrosine phosphatase. Similarly, phosphotyrosine components can be used as a module. Module means single functional unit. So only you take the domains and these can be used to build complex circuits or networks. Okay, so these modular, this is why domains are so important. You can take domains of a particular protein and it can function on its own. So it is a modular unit and you can incorporate this into another protein and then you can give this other protein the function that this particular protein had. Okay. I hope that is clear. Okay, now we will try to answer this particular question. Okay, try to answer this question. So option number A is it interacts with sequences containing the phosphotyrosine group, which is not true about the phosphotyrosine binding domain. Domain independent part. Yes. Domain independent part. So domain independent part is often the N-terminal and the C-terminal regions. These, I told you, these are also very important because you just saw this case of auto inhibition. So even the domain independent parts are important in the activity of the protein. Okay. Usually the domain independent part, this N and C terminal, they are very flexible. They are not very ordered, right? So it is kind of intrinsically disordered. Okay, so we'll have a look at this particular question. It interacts with the sequences containing the phosphotyrosine group. Okay, which is not correct. The insulin receptor substrate contains these domains. It is situated towards the C terminus. It has a compact beta-stranded sandwich capped by an N-terminal helix. So I am getting mixed answers. Let's see which is the right answer. So right answer is B. These are not correct about the phosphotyrosine binding domain. We know that phosphotyrosine binding domain, of course, interacts with the phosphotyrosine group. So 1 is correct. And we know that the fourth one, this 7-stranded beta sandwich structure, right? This one, it is there. And you have to know whether this is present towards the C terminal or the N terminal. That is something that you have to know. Okay. And so we see that this PTB is situated towards the N terminal and it has a compact beta-stranded, 7-stranded beta sandwich structure capped by the C terminal helix. Excuse me. So this is therefore the B, answer B is correct. Okay. Now we will look at some other domains. You already know these. So try to answer. One is a beta sandwich, another is a double zinc finger structure. In our last class, we talked about zinc fingers. So there is a double zinc finger structure. I can already see that. And there is a double hairpin structure. Which one do you think would be the right answer? Okay. Any other answers? A. Alright, let's see. The right answer is C, because see, you see these two zinc fingers here, and C will have double zinc finger structure, right? So anyway, A and B are getting eliminated. What happened? You guys are not concentrating. So A and D will anyway get eliminated because we can already see two zinc fingers. Zinc fingers means what? There is a zinc atom, zinc ion, which is coordinated by, let's say, 2 histines and 2 cysteines or 4 cysteines, right? That is a double zinc finger, which we can see in the third option. What about your double hairpin structure and beta sandwich? Beta sandwich we saw just now, right? What is there in beta sandwich? This is the structure of a beta sandwich. This one. There are 2 helices and these are the 7 beta strands, right? So you have 2 helices and 7 beta strands here. So this would be beta sandwich, that is C, and this would be your double hairpin structure. Okay. I hope that is clear to you all. Second is not hairpin. Yeah, second is hairpin. Second is double hairpin. First one is beta sandwich and the third one is your 2 zinc fingers. Okay. Right. Now we will talk about phosphomimetics. So phosphomimetics is a very, very important class of designing peptides. Okay, for example, let's say that a particular, let's say a protein, it is undergoing phosphorylation and it is performing a specific function. Now we will make a phosphomimetic. That means instead of that particular residue which can undergo phosphorylation, let's say we are going to introduce a particular residue where the phosphorylation, the group that undergoes phosphorylation is absent, it is not there. Then what is going to happen? This is how we can actually confirm the role of specific residues. Peptidomimetic. Oh yeah, this is peptidomimetic. I am telling you about phosphomimetic. This is very similar. So in phosphomimetic, this is focusing only on the phosphate group activity, but peptidomimetic means you can make different kinds of peptides using these. So see, peptidomimetics are small proteins like chain designed to mimic a peptide. So phosphomimetic is mimicking a phosphate group, right? Similarly, this is a peptidomimetic which is mimicking a peptide, and this is having particular molecular weight. So these are chemical structures derived from bioactive peptides. For example, this is a peptide that is shown, and the peptidomimetic is not having this CO group. Okay, so these can be of four different classes. They can be small molecules and they can be having peptide characters. So we will look at them in details. So this is how you design the peptidomimetics. So the different classes are shown here. So peptidomimetics involve strategic modifications to the peptide backbone, incorporation of non-natural amino acids. So what are non-natural amino acid? Non-natural amino acid means that a particular amino acid which is not found in nature. It can, let's say, be you take serine and no, not serine, because yeah, you take serine and, or let's say you take cysteine, right? And in cysteine, you know, this has sulfur. So instead of sulfur, if there is selenium, then it becomes selenocysteine. But selenocysteine is found in bacteria. But then, if it would not be found in bacteria, it would have been called a non-natural amino acid. So similarly, any amino acid that is not found in nature, that is called non-natural amino acid. And usually the stop codons are modified in such a way that the tRNA, there is no tRNA complementary to a stop codon, right? But then it will be modified in such a way that the tRNA will come and recognize the stop codon, and it will not treat it as a stop codon. Instead, it will treat it as a normal codon, and it will incorporate that non-natural amino acid in the position of the stop codon in the peptide. Alright. So this is how non-natural amino acids are incorporated. And there is cyclization of linear peptides. So these alterations are intended to enhance the molecule stability, optimize its conformation, and improve its interaction with biological targets. So there are different classes. The class A is modified peptides, primarily composed of proteogenic amino acids. So proteogenic amino acids are coded by genes. They are involved in formation of proteins and they resemble natural peptide binding epitopes. Class B, the peptides featuring a significant number of non-natural amino acids or substantial backbone modifications designed to mimic specific peptide conformations. Class C is making a scaffold, okay, to make a bioactive conformation of a peptide. Class D, the molecules that do not directly replicate the side chains or conformations of a peptide but instead mimic its mode of action. So these are the four different classes of peptidomimetics that you can make. There is one more different, okay, now how do we do that? So there can be backbone modifications, there can be side chain modifications, there can be cyclization that we saw in the previous slide. And another strategy is, this is structural modification. Another strategy is stapling. What is stapling? Stapling means staple peptides are a specific form of peptidomimetics where a hydrocarbon bridge is introduced to stabilize an alpha helix, enhancing the stability and target affinity. So this is, there can be hydrophobic staple. So hydrophobic staple means that 1 and the I plus 5. So I and I plus 5, these two positions on the alpha helix, there is a hydrophobic amino acid that is interacting with one another, and there is a side chain to side chain hydrophobic stapling. PPT not visible. Oh god. Okay, okay, 1 second. Okay, I hope it is visible now. Okay. Okay, give me a minute. I'll be back. I'll be back. Okay. Give me 2 seconds. Right. So I hope you can see the PPT now. Right. We'll quickly complete. Okay. We will quickly complete whatever is left. So we were talking about peptidomimetics and stapling. Stapling is very important when you want to increase the helicity of an alpha helix, increase its helical propensity. Right. Similarly, for other purposes, stapling is very useful. Okay. Now we will have a look at the questions on peptidomimetics. Try to solve this. Peptidomimetics are used for, one is identification of a tight binding potential peptide sequence, modification of the peptide to enhance stability, uptake and delivery, and development of drug-like compounds from existing peptides. Chalo, answer. C. Yes, yes, correct. C is correct because you can do all of the three using peptidomimetics. Okay, so peptidomimetics, identification of a tight binding potential peptide. How do you do that? You make a mimetic where you mutate the residues involved in the tight binding. Then you can confirm whether it is a tight binding sequence or not. The second one is modification of the peptide. You can always make modifications and you can improve the particular peptide. So, increase its stability, uptake and delivery. Uptake means insertion into the cell. Okay, transfection or transformation ability. And then the development of drugs, that is definitely there. So, it is all the three. And an example is given here. So in this example, you can see that a peptide is given and a peptidomimetic is given, and the peptide is nothing but somatostatin, and they have increased the stability towards proteolysis. They have improved the better transport properties or the cellular uptake and selectivity against non-target receptors. So by modification of that particular group, they have made these modified somatostatin, which is the right. So, and this is a hormone, right? And that is how they have improved its functions. Right. So we have one more question. This is on stapled peptides. Staple peptides bind to estrogen receptor using a hydrocarbon link to stabilize them in the form of a dash. What do you think it would be? Hydrocarbon link. A is beta sheet. Okay. Any other answers? The right answer would be alpha helix. We saw here that hydrocarbon stapling is assisting in the stabilization of alpha helices. Right. Hydrocarbon bridge is helping in stabilization of alpha helices. So this should definitely be alpha helix. right? Shape complementarity is required to interact for the interaction between the protein and the polypeptide. Okay, we'll go to the next question. Okay, we'll talk about renin angiotensin, and we have a few more questions left and we'll be done for the day. So during COVID-19, everybody was crazy about this angiotensin and renin interactions, right? So angiotensin and renin is a very important interaction, and this is helping in maintaining your blood pressure, vasoconstriction, vasodilation. So these receptors that you're seeing, MAS and ATI are it, yeah, so these 81 are, so these are actually receptors, and this renin angiotensin mechanism, this is actually in helping in different processes such as vasodilation and vasoconstriction, nitric oxide, oxidative stress, inflammation, fibrosis, proliferation, etc. So right, so in the renin angiotensin system, ACE2 and angiotensin, see 1 to 7, this one ACE2 is the receptor, sorry, ACE2 is here, and it is basically, so the protease renin converts angiotensinogen, which is a proenzyme, angiotensinogen into angiotensin 1, which is subsequently converted to angiotensin 2 by angiotensin converting enzyme or ACE. Okay, so angiotensin 2 can bind to angiotensin type 1 receptor or AT1R to exert actions such as vasoconstriction, hypertrophy, fibrosis, etc., oxidative stress. ACE2 is another enzyme which is converting angiotensin 1 to angiotensin 2, and the number of amino acids is also given. Angiotensin binds to the MAS receptor to exert action such as vasodilation. So exactly the opposite to what was happening in the AT1R receptor. So angiotensin 2 can also bind to AT2R to counteract the aforementioned effects mediated by AT1R. Okay, so these are actually opposing each other's functions. So now you try to look at this question, which is incorrect about ACE inhibitors. So it produces angiotensin 2 that causes blood vessel contraction to increase blood pressure. Angiotensin 1 is a hormone that is converted from angiotensin 2 by the enzyme ACE2. ACE is a highly specific carboxypeptidase. So to understand whether this is a carboxypeptidase or not, you need to look at this diagram. This is why I have attached this diagram here. So where is it making a cut? So for that you need to see the sequence, right? This is 1 to 9, histidine, proline, phenylalanine, histidine. Again histidine, proline. So that means the cut is here at the C terminal. And so the carboxypeptidase means that it is going to make a cut from the C terminal. So now you tell me which one should be the right answer. C. I went to that slide by mistake. Okay. Okay, great. Yes, C is the right answer. So angiotensin, which is incorrect. Both 1 and 2 are incorrect because C. Okay. So these two are incorrect. Angiotensin 2 is a hormone that is converted from angiotensin 1 by the enzyme. So it is not the other way round. It is rather from 1 it is getting converted to 2. Okay, by ACE. And the last two residues of angiotensin 1 are cut off by ACE enzyme, which is a carboxy. Yes. Yes, tell me. You have anything to say? Yeah, sure, you can unmute. It's raining outside.
Can you hear me, madam?
Sorry?
Hello.
Yes, ma'am.
Can you hear me?
Yes, yes,
Question was which among the following is are incorrect about ACE inhibitors?
yes.
So it produces angiotensin 2 that causes blood vessel constriction to increase blood pressure.
Hmm.
So ACE causes conversion of angiotensin 1 to angiotensin 2, not ACE inhibitor.
Yes, that is correct. So ACE inhibitor is not going to cause that, but the enzyme is going to cause that. Right.
So that's why one is incorrect. Angiotensin 1 is hormone that is converted to angiotensin 2 by enzyme ACE, that is correct. And ACE is highly specific
This is
carboxypeptide.
correct.
You had asked incorrect, not the correct.
Ha. So this is wrong. Right. 2 is wrong. Because 1 is not converted from 2 by ACE. But 1 is
Okay.
converted to angiotensin 2 by ACE. Got it? Here they
Okay,
are
okay.
saying that 2 is converted to 1. That is not the way it works. It is from 1 to 2.
But
That is one
one
thing.
is also incorrect, madam.
1. Right. Let's see. It produces angiotensin 2 that causes blood vessel contraction to increase blood pressure. So when we look at angiotensin 2, right, that is what they are asking in the question. Angiotensin 2. Yes. Angiotensin 2 is causing vasoconstriction, which is increase in blood pressure. And
No, that's correct. That's correct. But it is not produced by ACE inhibitors. It is produced by ACE only,
hmm,
not by ACE inhibitor.
right. So then inhibitor should
Many.
not be there in the question.
That's what I am asking.
Okay. Then you
So
can
if
write
inhibitor is
the
there,
yes.
to whom we should write, madam?
You should
Too
write to
many wrong
NPTEL
answers
team.
are there. Okay, madam, you continue. It's
Okay.
okay.
All right. Let's continue and let's finish up quickly. Okay. Right. So this is a question about molecular chaperones, and I hope you remember anything about chaperones, something at least. So chaperones are proteins which we had studied previously, and they are useful in folding proteins, right? Folding the newly synthesized proteins. So see which of them can be correct, and we can have a look at these. So it belongs to a large family of molecular chaperones. It has a key role in protein. This is kind of a match the following. So it has a key role in protein maturation, refolding and degradation. It is involved in protein folding in response to stressful conditions such as exposure to high temperature. So if you remember the heat shock proteins, they were very useful in folding of proteins on exposure to high temperature. It is involved in folding of signal transduction proteins. Okay, so we have got one answer. Let's look at the answer that they have given. So first 3 points are the features of HSP. HSP meaning the heat shock proteins, and the fourth one is specifically for HSP 90. So HSP 90 is the correct option. I am not sure what the question was, but then you can just keep this in mind that these three are basically corresponding to HSP 7 HSPs in general, and for HSP 19 it is useful in folding of signal transduction proteins. Okay, yeah. Now we will have Now we'll talk about chaperonins, because for the next question that is important. So chaperonin, you know that these assist the chaperones in protein folding. So one example is given here. This is found in bacteria, the GRO-ES, GRO-EL and the GRO-EL again. So there is one cis ring and there is one trans ring, and the unfolded polypeptide kind of goes in. It is like a cylinder and the cap closes. The cap is your GRO-ES and the cap attaches, causing the cylinder to change shape in such a way that it creates a hydrophilic environment for the folding of the protein. The cap comes off and then the protein is released. Okay, and here the substrate is bound in an encapsulated way, and this is the bound substrate that we are seeing. So this is all about chaperonins. It's a hollow cylinder. Now you try to answer this question. There is one grow ES, there is grow EL, and cis and trans. That is all you need to remember. Come on, it's easy. Yes, right. So protein belongs to chaperonin. The upper part is grow ES, this is your grow EL cis, and this is your grow EL trans. Now we'll go to databases. This is the last topic for the day. So databases are very important when it comes to bioinformatics studies. Okay, databases can be primary databases, there can be secondary databases, there can be different kinds of databases, and there are so many databases now. For DNA, for proteins, there are separate databases. Okay, so one example, what is a primary database? So primary database means whatever information you get about a particular protein or about anything at first initially, you take all that information and you dump it somewhere. That is your primary database. And what about the secondary database? So in secondary database, this is more organized. This takes one particular protein and it tells you about a lot of things about that particular protein. The secondary database is containing information derived from the primary database. Okay, so primary database can be sequence database and structure database. Example of structure database is protein data bank. Other examples are given here. Your for DNA. Okay, so for let's talk only about proteins here. So for proteins, the structural database is protein data bank. Okay, and for the protein sequence, you can go to GenBank. GenBank is telling you about DNA as well. You can go to DDBJ. J stands for Japan. So Genomic Data Bank of Japan, that is also telling you about the sequences. So this is a sequence database for DNA. Okay, now we go to the secondary databases. So secondary databases, for example, UniProt. UniProt is very widely used. There can be, this is UniProt is actually a knowledge database. So knowledge database means it's giving you information about everything of the protein. The cellular localization, the different domains present, whether the protein is interacting with other proteins or not. Very little information, but then yes, and whether what are the different isoforms of that protein, what post-translation modification it is going through, what functions the protein is having, everything is given by UniProt. Now we come to string database. String database is something that gives you information about the entire molecular network of interactions of a particular protein. Okay, so let's say you choose one particular protein and you click on that protein and it will give you like a full network of all the different other proteins that this particular protein interacts with, whether it is activation that is happening because of this interaction or repression of particular other function, everything will be given by string database. Similarly, there are structural databases, for example, Cath and SCOP. You will get questions from Cath and SCOP. So Cath and SCOP are structural databases which tell you about the different structure of the proteins, the families into which these structures come, whether these are all alpha proteins, all beta proteins, alpha plus beta, okay, and then whether it is having a, let's say, beta meander, whether it is having a beta sandwich, everything will be told by Cath and SCOP. Okay, now we will, and then there is also other databases. For example, you have this string comes under XPCE database. Okay, so XPCE is a huge thing and under it comes different other things. So yeah, that way you can get to know about the PI of the protein, you can get to know about the molecular weight of the protein, everything, functions of the protein. Okay, so yeah, now ITAZR and FIRE2, these are predictive databases. Similarly, AlphaFold is there now, which is very useful. So ITAZR, FIRE2, these are all predictive databases which give the predicted structure of the protein based on different methods, which can be sequence homology, which can be other methods. Okay, right. Okay, so these are some examples that are given. This is string database, this is UniProt, this is ITAZR. You can have a look at them. And finally, we come to the question. This is the last question of the day. So, let's try to answer this question now. Choose the correct statement about the database of interacting proteins. The secondary databases combines experimentally inferred data from the primary database with computational predictions of molecular interactions. The predictive databases combine experimentally inferred data from the primary database with computational prediction. Right. The predictive database combine experimentally inferred data from the secondary databases with computational prediction of the molecular interactions, and the secondary databases combine the computational prediction data taken from the predictive databases with experimentally inferred data of the molecular interactions. So the right answer is B. The predictive databases take experimentally derived data from the primary databases. So remember, secondary database, they are always taking data from several primary databases. Primary database is taking information mainly from experimental data, and predictive databases are performing computer-based predictions and combining that with the data from the primary databases to give you information about the particular protein of choice. Okay, thank you so much. These are the different references that you can go through, and thank you for your time. Thank you for your patience. How is A not correct? Okay, we go back to A. See, secondary databases combine experimentally inferred data taken from primary databases along with predictions of molecular interactions. No, secondary databases take information from different primary databases. So whatever is given in the secondary database, it is not a predictive thing. It is a proven, experimentally proven. There are even references of the papers, published papers from which they have derived that data. Okay, and that is why this is not predictive, but this is proven. They may give you structures from AlphaFold, but it will be mentioned there as AlphaFold. Okay, there will mainly be structures from PDB before AlphaFold. If they are not structures, then they'll include AlphaFold structures. But the function is basically to give information about a particular protein, all the information that is available on the internet but published. Got it? Thank you so much. Okay. Thank you. If there are no more doubts, we'll end the class. Thank you so much. Thank you for your time. Thank you for your patience.